I'm Michelle Hudson, a research project manager. I've had the good fortune to run a variety of complex and interventional clinical studies over the last 12.5 years for internationally recognised researchers such as Professor Frayling, who's talking to you today. Also in our audience are members of the DIVA and FATDIVA study teams. There's Andy Pitt and Lindsay Beale, our senior research nurses, and Becky Dobbs, our graduate research assistant, who is invaluable. Becky and Peter Hollingsworth have done a great job helping to arrange this event, so thanks to them. So a warm welcome to you all. It's important for research participants to receive feedback about the research they've been involved in. You kindly participated in either DIVA or FATDIVA, which are short, memorable names of the two studies that Professor Frayling will be providing more detail about today. So huge thanks for joining us today. This is actually the first online feedback event that we've done for a CRF-run study. We hope it'll prove to be a successful, easily accessible and interactive format that is enjoyable for you and could be adopted by future studies. We do recognise that a webinar doesn't suit everyone. So we'll also create a newsletter which we'll send out and upload to the research feedback pages of the Exeter CRF website. If you've not visited our website before, just typing Exeter CRF into your search engine should find it. So a bit of housekeeping. Firstly, be aware that this event is being recorded. We will upload the recording to the CRF website. To preserve bandwidth for a clear presentation, we've turned everyone's camera off and have muted your microphones. For those of you not familiar with Microsoft Teams events, in your top menu, there's a chat icon, which if you click on it, will enable you to type your questions. We aim to answer questions at the end of each study section and at the very end of the session, so please type any questions you have into the chat. But if you would particularly like to ask your question by speaking, please could you either indicate this with your question in the chat or use the raise hand function, which you'll see in the menu. As this is our first feedback event, we would be very grateful if you could complete a short questionnaire and a link will be emailed to you after the session. This will only take a few minutes to do, but it will help us to gauge your experience of the webinar and to collect your feedback and suggestions that could improve how we run future events. So you've all visited our research facilities within the Research, Innovation, Learning and Development Building, otherwise known as RILD, at the Royal Devon and Exeter Hospital. We're really proud of our outstanding facilities and you will have seen our bespoke wards, but we also have consulting rooms and sample handling laboratories, plus we have specialist equipment for investigating exercise, physiology and metabolism, and also have access to the Muriel Gillings Neuroimaging Centre. The Exeter CRF was set up in 2008 to run experimental clinical studies, and it is a partnership between the Royal Devon University Healthcare NHS Foundation Trust and the University of Exeter. It is one of 28 clinical research facilities across England whose infrastructure is funded by the NIHR. That's the National Institute of Health and Care Research, the British government's major funder of clinical public health, social care and translational research, which was founded in 2006 and has a mission to improve the health and wealth of the nation through research. To cover additional salaries and consumables, we're reliant on additional funding from external grants secured by researchers to run specific studies. Professor Frayling is the chief investigator of the two externally funded studies being discussed today, and his funding contributed to the salaries of highly trained research-dedicated nurses, scientists, doctors, technicians, and support staff like me. Another resource that the Exeter CRF is proud of and that you're all part of is Exeter 10,000, often referred to as Extend. This is a study and research resource set up to act as a gateway to further studies. Anyone over the age of 16, with or without health problems, can take part. So if you have any friends or family that would like to take part, do please ask them to contact the CRF. Our contact details are on the CRF website and at the end of this presentation. Our original aim was to recruit over 10,000 amazing local people like yourselves who are willing to provide blood and urine samples, body measurements and answer simple health and lifestyle questions. Most participants are happy to be contacted about future projects that they may be suitable for, making Exeter 10,000 a very helpful recruitment resource that has contributed to numerous important research studies aiming to increase our understanding of common diseases and healthy ageing. The samples and health information from Exeter 10,000 and other studies are stored securely in the Peninsula Research Bank, known as the PRB, which is a tissue bank with NHS ethics, governed by a steering committee made up of scientific and public members who carefully review research applications to make sure they're appropriate and scientifically robust. So, having covered the purpose of the Exeter CRF, the Exeter 10,000 study and the PRB, this leads me to explain that Professor Frayling applied to the PRB and was granted approval to approach you to invite you to the DIVA and FATDIVA studies. We thank you profusely for agreeing to take part, and I'm now delighted to introduce Professor Tim Frayling. Tim has been working as a molecular geneticist for over 20 years. In 2007, he obtained a personal chair as Professor of Human Genetics at the University of Exeter, where he heads a team of 14 that has become internationally recognised as a world leader in the genetics of common human traits and diseases, particularly type 2 diabetes, obesity and related conditions. So it's over to you, Tim, to feedback the findings of the DIVA and FATDIVA studies. We're all interested to hear why people put fat in different places and what it means for our health.
Thanks very much, Michelle. Just give me a moment to get some slides. So I have some slides basically to take you through what we've been doing. But firstly, let me just reiterate what Michelle has pointed out. This is also a fantastic opportunity to say thank you to all of you for volunteering your time and energy, often blood samples and various other things about you that will help us do world-leading research into genetics and diabetes and related traits and approaches to tackling disease at the Exeter Research Institute. And also a thank you to Peter and Becky for arranging this webinar. Quite a lot goes into arranging this. Okay, so as Michelle kindly pointed out, I'm going to talk to you about two studies that have involved you and recruiting you and measuring you in various different ways at the clinical research facility after you kindly volunteered to be part of the Exeter 10,000. These two studies, the first of which actually started as far back as 2014, and that timeline kind of reflects the extent of the work that goes in to recruit people into research studies, to perform the research, to analyze the data, all whilst we're doing lots of other jobs, such as, in my case, teaching undergraduate students and postgraduate students during the COVID epidemic, for example. So I think we're slightly behind schedule in terms of feeding back to you, but hopefully we can make up for it today. So the two studies have something in common, and that is that they are both about trying to understand why some of us develop type 2 diabetes and related conditions, especially in a world where it's hard to stay slim, given all the food around us and the ease with which it is to not exercise. And so I've kind of slightly tongue-in-cheek put the title up here as How to Avoid Type 2 Diabetes in a World Where It's Hard to Stay Slim. And of course, these two things are largely beyond your control. But one way is to have more or better functioning pancreatic beta cells. And these cells are the very, very special cells that exist in about less than 1% of your pancreas. This very important organ that sits next to your gut and is largely responsible for secreting enzymes and molecules that break down all your food into sugar and digestible nutrients. But These special cells in the pancreas, called beta cells, are the only cells in the body that produce insulin. And insulin, of course, is the only hormone that reduces the amount of sugar you have in your blood and controls the amount of sugar you have in your blood. Sugar levels in your blood are a bit like body temperature. We need to keep it very stable at a very set level within a narrow range. There are lots of hormones which keep your sugar up if it goes too low, and that's because it's very dangerous if your blood sugar goes too low, because that can damage the brain. But there's just one hormone, insulin, which actually lowers blood sugar levels. And that's important because if your sugar remains too high, it damages you, especially your blood vessels. And I'm sure many of you have heard of the complications of type 2 diabetes, such as having damage to your eyes and your kidneys, etc., as well as your heart. So to avoid diabetes, it's good to have lots of pancreatic beta cells that are producing lots of insulin. And another way is to actually have better functioning fat cells. So this is something that's perhaps not quite so familiar, but if you have more and better cells under your skin, what we call subcutaneous fat cells, rather than around your organs, around your pancreas and liver and heart, then that's a better place to put your calories, and especially your excess calories, excess fat, if it's been hard to stay slim. And some people are better at storing their calories under their skin than around their organs than others. Most obviously, women are better at doing this than men. Women tend to have more fat overall, but they tend to, compared to men, but they tend to put it under their skin or in their lower body, whereas men tend to have more fat around the organs and in the upper body. But nevertheless, there's great variation within men and within women about where the fat goes and how much insulin we secrete. So that's the sort of starting general premise. I'm now, let me see if I can move my slides forward with that arrow. Yeah, there we go. So the next handful of slides are setting the scene about genetics. So the way we have tackled the questions about making more beta cells and making more fat cells is to take a genetic approach. So my team and actually many people at the medical school in Exeter, experts in understanding how the DNA sequence and the genes influence our bodies. And the genes can be thought of as the architect's plans for the building of a body. So your genes don't actually contribute to your body. So the genes are like the architect's plans for building a house, and we have about 20,000 genes. So that's 20,000 drawings, if you like, to build our body. Instead of drawings, of course, what those architect's plans are is a code, a long string of A, C's, G's, and T's for the 4 molecules that make up our DNA. And it's just that instead of having a few drawings to build a building, we have 3,000 million of these letters, so 3 billion letters. And in many ways, genetics is very simple, because you can think of our DNA sequence as this just one long line of various sequences of these four letters, but 3,000 million of them. And we have about 20,000 sections of those plans, that code, which separately encode the proteins. And in this case, I've got the protein insulin, where the code tells the body to make insulin. And in other genes, the code will say make a particular protein for a muscle cell, or a particular protein that's important to a brain cell, for example. And those 20,000 different items, different genes is how we build our body. The slight complication, of course, is that we have 2 copies of the architect's plans. We have 2 copies of the genome, which means we actually have 6 billion letters of the gene code, because we inherit 3 billion from mum and 3 billion from dad. So we have these 2 copies sitting side by side each other. And your mum's copy is actually a kind of a shuffled up mixture of her parents' genomes, and your dad's copy is a shuffled up mixture of his parents' copy. So your grandparents' copy is all mixed up, shuffled up a little bit like a deck of cards. And the important thing here is that as well as genes, we have variation in those genes, and this is what enables us to look at differences between people and equate the differences between the genetic code to differences in people. So what you can see here is that mum's DNA code is almost identical to dad's DNA code, but there are in 3 places a difference. So you can see the 6th letter along here is in mum is a C, as the 6th letter along here in dad is a T. And I've represented this block as that might be encoding a gene. So this gene, when inherited from dad, might look a little bit different to the one inherited from mum. And so what that means is we end up with a house encoded by mum's copy and dad's copy, but there might be some subtle differences. And here's another example where, you know, the insulin molecule encoded by mum's copy of the genome might look a little different from the copy encoded by dad. Usually, you know, those copies, or often those copies can work in just the same way, but sometimes there are subtle differences, and those subtle differences in how the proteins function and when they're switched on and off contributes to the variation in how our bodies look and function. I'll just skip over that one because I've just mentioned those. Importantly, this means for any one position in the genome that varies, there's actually sort of 3 possibilities. So if you have a position in the genome where there's a C in mum's copy, there's a C, but in other copies of the genome there might be a G. That means you could actually inherit a C from both mum and dad. You could inherit a C from mum and a G from dad, or you could inherit Gs from both mum and dad. So we can, for any one variation in the human genome, we can kind of divide people up into three groups according to the number of C or G variants we have. And all we do in our genetic studies is take each one of these variations in the genome and we try and ask the questions such as this. If people have more copies of the C or less copies of the C here, do they have differences in the things we're interested in? Are they taller or shorter? Do they have higher or lower BMIs, higher or lower risk of diabetes? Does it change eye color? And in this case, does it change number of beta cells? And pretty much anything you can think of that varies between people will be influenced by at least some genetic variants and changes to this DNA code. So you are now all experts in human genetics. You've passed your introduction to human genetics, and I hope that basic introduction will make the 2 presentations about the specific studies a little easier to follow. For each of 2 presentations, I am going to split them into roughly 4 sections. And those 4 sections basically form the way we do science, the way we do investigations. And again, really pretty simple, really. We start off with a question, and we try and decide whether or not that question is important or interesting, or preferably both. And so we start off with why did we start? And that usually ends up being the introduction to a scientific paper. And then what did we do? Ends up being the methods. What did we find? Ends up being the results. And then what does it all mean? It becomes the discussion of our scientific findings. So why, and another general question, why did we start? So why do we study genetics when these are the genetic variations and changes in your DNA sequence that you inherit from your mum and dad, and so you can't change it. We can't go in and sort of modify the DNA sequence in a robust way as yet, anyway. And so why do we study it? It's not like measuring your cholesterol level and then giving you a drug to lower your cholesterol level. And there are several reasons, and this would be a separate talk in itself. But very briefly, it basically helps us understand how the body functions, and that can spin off into lots of new research, and more particularly can help us understand if and how particular therapies, particularly drug therapies, might work. And then the second reason is we might identify, second broad reason is that we might identify different groups of people to help more personalize your treatment. As I say, those are sort of separate topics in themselves, but suffice to say, lots of good reasons for why we want to understand how the DNA influences our bodies. Okay, so I am now going to talk specifically about the first study, which was called DIVA, shortened from diabetes variants. The variants referring to the genetic variation in our DNA code that I spoke about. And this study, we decided to look in more detail at one particular gene that's called CCND2, and a particular version of this gene, where a variant in the gene which was a C rather than a G, had previously been shown to greatly reduce the risk of diabetes, greatly reduce the risk of type 2 diabetes, and by about half. So this variant is present at half the rate in people with type 2 diabetes compared to people with type 2 diabetes. So it's kind of a protective effect. Really interestingly, these people who had a lower risk of type 2 diabetes because they had a copy of the C variant were also taller. And this fitted with what we knew about the gene from other studies, that it's critical to growth. And that data came from, and that insight came from, in particular, studies in mice. So previous researchers had looked at what this critical gene was doing in mice and shown that if you have mice that lack this particular gene, CCND2, they are smaller, they have fewer beta cells, and they produce less insulin when given sugar, given a sugar feed or injected with some sugar. Their pancreas responds, their beta cells respond by producing less insulin than you would like. And what you can see here on the left is a stain of a slice of pancreas showing the islets and the special parts of the pancreas that contain the beta cells and the insulin producing cells, stained in green here. And then on the right, you see on the second picture here, you see that the mice without this gene have much smaller numbers of beta cells. And on the right, you can see that translates to the mice without the particular gene responding differently to a sugar challenge. And what this thick line here shows is that their sugar, when given a sugar challenge, when injected with sugar, their glucose levels stay high for much longer, go higher and stay higher than the mice who have the gene, suggesting these mice are what we call glucose intolerant and are basically more likely to be diabetic. So all of that information made us think it would be interesting to study people. Now, of course, we have a problem. It's hard to measure the number of beta cells you have. The only real way of doing this directly in people is sadly to wait until they die and take a sample from an autopsy. And that is possible, and that has been done in relatively small numbers of people, and we have experts at Exeter who do a lot of that work. But when you're studying particular genes, some of which might not be very common, that's not a very practical way of doing things. And so what we decided to do was look at a proxy measure of the number of beta cells. And that is something called a glucose potentiated arginine insulin secretion test, which is a bit of a mouthful. But this is a way of, and this is what you or many of you were recruited to and came into the clinic to undertake. This is basically a way of testing out your beta cells, putting them through their hard yards, if you like, to see how much insulin they can really secrete when really pushed. And the way we really push your beta cells to produce as much insulin as possible is as follows. So for those of you who did this test, you'll remember coming into the clinic and having a cannula in your arm, which was primed to produce, to infuse glucose. But before we did that, in your other arm, we gave you a small injection of a substance called arginine. And arginine goes to the beta cell and basically tells the beta cell to produce insulin, start releasing insulin. And we did that test at the start, and we measured how much insulin you produced at the start. And then after we'd done that, we kept you wired up for 10 minutes. And then we started injecting gradually glucose, sugar, and so that we took your blood sugar levels up for about half an hour, up to quite a high level, a sort of level that you wouldn't want to be staying there for a long time. But we took it up for just half an hour. And then when your glucose levels were at about 20, so about 4 times normal, we gave you a second injection of arginine. And that's really putting your beta cells under the test and saying, right, let's see how much they can really produce when they're being challenged with sugar and arginine. How much insulin can you produce at sort of maximal stimulation? And then we allowed you, after that, we allowed you to recover and have something to eat, check you're okay, and went home. And this was a complex and intense protocol in the lab, particularly for Andy and Lindsay and others who did this, because at times they were taking a blood sample every 2 minutes from your arm at very specific time points. Because what happens when you have sugar and arginine injected into your veins, it goes straight to the beta cell, and you produce insulin almost immediately. Whereas normally, when we measure people's response to insulin, we give them a meal, we give them sugar, and it goes to the gut. And so it's a lot slower process because the sugar and the food has to be processed through the gut first. So this all happens much quicker and is also a more direct way of measuring the function of your beta cells. And we think that's also a good measure of how many beta cells you have. So what do we do next? Let me just move this forwards. Yeah. So the people with one version of the gene that protects you from diabetes, or the gene that's in fewer people with diabetes than people without diabetes, is only present in 1 in 25 people, which is where the sheer number of people in the Exeter 10,000 study became very useful, because it meant we could actually have about 260 people carrying this particular protective version of the gene, and then we could match them to people of the same age, sex, and BMI, so that we took out any variation in these measures that might be due to getting older or being a man rather than a woman, or being a bit fatter or thinner compared to other people. So this is where Michelle and her team came in, and they spent a lot of time matching people up in the database who were basically the same sex, very similar age within 2 or 3 years, and a very similar body mass index. That made it very tough because sometimes if someone dropped out of a study or the test didn't work, we had to go back and find somebody else with the same characteristics, which is another reason why the study took quite a long time. But in the end, we ended up with, and I won't go through all this, but basically, to cut a long story short, we ended up with 51 pairs of people, so 102 people, where 51 had one version of the gene and 51 had the other version of the gene. And so we did those 102 tests. All of those people, actually, it ended up being more than 102 people, because sometimes we couldn't get people matched up, but we ended up being able to analyze data from the 51 pairs with this particular test. A shout out to the CRF and the other people involved. I've mentioned Andy and Lindsay and Michelle. Andrew and Angus were our clinicians providing me with clinical support, and Tim and Rachel from the biochemistry lab did a huge number of insulin and glucose tests. So on to the results, basically the bit where what did we find? And these graphs, I'm just going to divide it into sections where we show what we found in general and then what we found in relation to the gene. This just is a confirmation that, with one exception here, we did a very good job, or Michelle did a very good job at matching people according to their BMIs and ages. So for example, here at the top, you can see that somebody with one version of the CCND genotype who had a BMI of about 35 was matched to somebody else, the different genotype, but with a BMI of just under 35, etc. And here's somebody who came into the clinic with one version of the gene who was about 73 years old, and they were matched to somebody else who was 73 years old. So those nice horizontal lines show that we did a good job at matching people up. So as I say, I'm going to, the next few slides are the results, and they are divided into what we found in general and then what we found when we compared the two groups of people based on the genetic variant in this particular gene. And this slide shows the first slightly surprising result, I think, in that, as expected, we showed that when their beta cells were stimulated with the arginine or the glucose, they produced more insulin. What we didn't expect was quite how much variation there was in those measures. So this graph with all these colored lines on shows on the horizontal axis, it shows what happened to your insulin, and each line on the graph is an individual person. So if you're in this study, you will be one of these lines. I haven't attached your names to them in the interest of confidentiality, of course, but some of you were producing huge amounts of insulin at this time point, and others fewer. So x-axis, the time point during the study, y-axis, vertical axis, the amount of insulin you're producing. And you can see at the start of the study, at the start of the test, when we just gave you arginine, you all produced more insulin, sometimes up to 1,000 picomoles per liter, others less. And then you went back down again, and then glucose started to be infused. So you start to produce insulin to try and combat the amount of sugar that we're giving you, and so your insulin levels go up. But then this is the bit where we really test out your beta cells by giving you a second bolus, a second shot of arginine, and this is when your beta cells with their insulin really go poof, and I need to produce all of my insulin. And you can see though that whilst everybody certainly did produce more insulin, there's a huge variation, and some people seem to be able to secrete a huge amount compared to other people. And that's actually the same data, but summarized in in a slightly more scientific way. So you can see that the line on these bars is actually the average amongst all the 102 people of how much insulin, and then these bars are the kind of range of insulin that was produced. So the critical results are that what we noticed, again, slightly unexpectedly, because we were hoping that your body mass index was kind of taken out of this equation, but we showed that people who had higher body mass index had a higher response, produced more insulin. We think that is a kind of a proxy for having more beta cells, or needing to have more beta cells and ability to secrete insulin, because people who are a bit more overweight and have higher BMIs but don't have diabetes kind of need to produce more insulin to stay non-diabetic, and so that does make some sense in that people, as you can see, people overweight, more overweight here, were producing more insulin when we were stimulating their beta cells. When we look at the results where we compare the two groups of people, those with and without the rare variant in CCND2, we confirmed that there was an effect on growth, and would have been an effect on growth in childhood, in that the people carrying the alternative version of the gene were on average 4.7 centimeters taller than those without the gene. So the people who were taller, they're the ones with the protective allele for diabetes. So in old money, that's just under actually a 2-inch kind of difference in height, which is quite substantial considering that this is just one change in your DNA sequence from amongst 3,000 million. That was nice because it confirmed that what we were looking at made sense. It was associated with growth, and therefore, could be associated with the number of beta cells you had. It meant that we set the study up correctly, and everybody being recruited correctly, a sort of positive control. What this slide shows is then splitting people up and how much into the two groups, and showing how much insulin they were producing at the different time points. And what we found was that there was no difference that we could find anyway between the two groups of people. So the people with the protective version of the gene were producing, which is in gray, the gray line here. This is the amount of another measure of the amount of insulin they're producing after we've stimulated their beta cells. You can see that the people in gray are the ones with the protective version of the gene, and the ones in black, people without the gene. There's no difference between the lines, so we didn't see any difference at that first time point. And similarly, even when we stimulated the beta cells, your beta cells, with the maximum stimulation possible, so the sugar and the arginine, that even though you were all producing lots more insulin, on the vertical axis here, there was no difference between the grey and the black lines, the two groups of people. So unfortunately, we didn't see a difference between the two groups of people, which meant we were unable to say this gene is important in the number of cells you produce, the amount of insulin produced, and therefore likely the number of beta cells you have. This is another plot of the same data, and this is actually plotting two measures. And this time on the horizontal axis, we have a measure of your insulin sensitivity, which we're also able to measure. So basically, how does your body respond to insulin? How quickly is it able to get rid of the sugar from your blood? And this is a very typical and famous pattern you see when measuring people's insulin sensitivity. So basically how your muscle and liver take up glucose compared to how much insulin you produce on the vertical axis. And the people who are very sensitive to insulin, they're very good at taking up sugar and reducing the amount of sugar in their blood. And muscle and fat cells and liver cells are working very well in that respect. They need to produce less insulin, so they produce less insulin here. And whilst the ones which are not very good at the people who are less good at taking up glucose need to produce more insulin up here to combat the high glucose levels. So you see this particular sort of curved shape graph, which was very nice to see. But you can see in the black squares and the triangles, those are the two groups of people based on the gene, and you can see there's not really a difference between the two groups of people. And that's the similar sort of data at that time point. So what does it all mean? It means that we didn't find any evidence that people with different versions of the gene had different numbers of beta cells, at least based on that measure of their ability to secrete insulin, which means that maybe the gene doesn't work that way. Another possibility is that we just didn't have enough resolution, didn't have enough power to see that difference, and maybe we needed to do the test in even larger numbers of people. So I'm going to stop that part of the presentation and pause to see if there are any questions. Okay, we're up to an audience of 58, which is great to see so many. Okay, we have some questions coming in, which is good. So to answer Basil's question in part, so arginine, the role of arginine is well understood. It's known that it's an amino acid, so it's a small chunk of a protein. So and it's known to be what we call a insulin secretagogue, which means that when it touches the beta cell, it responds because it's a nutrient, and the body's response to nutrients is to produce insulin, start producing insulin to ensure that your blood sugar and nutrients don't go too high. We've known about that one for a long time. Your other question, Basil, about CCND. So one thing you might have spotted is that we didn't look at people, or we didn't really have enough people who had 2 copies. So if you remember, 2 copies of the rarer version of the gene, because that would only be 1 in 625 people, and so we didn't have enough people to say, well, if you've got double the number of versions of that gene, are you 9 centimeters taller, for example? Because I think we only had 1 or 2 of those individuals, so statistically we wouldn't have been able to do that, and I think we'd have needed studies in the order of several hundred rather than 50 pairs to be able to answer that one. But we would kind of, generally speaking, the more number of copies you have, we would expect an effect on the measure, such as height. And then there's a question about adverse effects of arginine. Some of you may have noticed that when you were given the arginine, it produced a funny taste in your mouth. So someone did the test. Andy did the test on me as well. So you'll be pleased to know that, as well as asking you to take the test, I put my money where my mouth was, and Andy did the test on me. And I remember having a chalky taste in my mouth with the arginine, which is something to do with the effect of arginine on the cells in your mouth. But we don't exactly understand why that happens. Okay, I think we should move on. Do keep thinking of questions, because I think with a bit of luck we should have a little bit of time at the end. But I will now go on to FATDIVA. So this study, hopefully your slides are coming up. So the second study is similar in principle, in that we were trying to understand how genes might affect a type of cell that should help you avoid diabetes. But instead of beta cells, which produce more insulin, This one was about fat cells, particularly subcutaneous fat cells. So this, the fat cells that are under your skin, how they cope with excess calories and how they are able to store this molecule called triglycerides, which essentially is the scientific name for fat. And it's much safer and better, as I say, to store it under your skin, around your liver, heart, and pancreas. So over the last few years, our team, particularly Hanya Yukutka and Susie Martin, and several of us working over the past 9 or 10 years, actually, together with other groups around Europe, have begun to understand the genetic component to different types of fat. And what's become clear is that some genes are making us a little fatter, but in a healthier way, and we've called this favorable adiposity. And by favorable, in green here on the right, that means that you might be a little fatter, but you are better able to respond to insulin, so you have what we call lower insulin resistance, you have a lower risk of diabetes, a lower risk of heart disease, a lower risk of hypertension, high blood pressure, and you have healthier blood. Basically, you have less fat in your blood, you have more good cholesterol and less bad cholesterol, and various other markers in the blood of metabolically. Whereas in red, you can see that there are people and genes and factors which mean you put on weight, you end up with more insulin resistance. And this is the more normal accepted thing, that as you put on weight, you're more likely to be insulin resistant. You have a higher risk of these conditions and unhealthier blood, basically. And one of the critical factors that you can see in this little cartoon, we think, is that that means more of your fat is in and around your organs and in the upper body compared to under the skin and in the lower body. And Hanya and Susie and others have identified some of the genetic reasons behind these differerences. Only accounts for some of it, but it's really interesting in that you can see 2 types of genetic variant here. This time I've labeled both 2 genes, both of which have 2 versions, which I've just called A and B for simplicity. There's one which has a very traditional pattern in that the B copy of the gene, if you have a B copy of the gene, or if you have a B copy of the gene, you have higher body fat percentage, and if you have 2 copies of the B version, you have an even higher body fat percentage, and you can see that you also have a higher risk of type 2 diabetes. That's kind of what we'd expect. But it was really interesting to show that some genes did the opposite thing, and that whilst the B copy of the gene, in this case, in a special gene called PPAR gamma, PPARG, was going with higher body fat percentage, those people on average had a lower risk of type 2 diabetes. Fewer of them had type 2 diabetes, which was really interesting. And so we decided, and Hanya and Susie decided to look for more of these genes, and they did that by looking at large amounts of data from other studies, sometimes tens of thousands of people, looking for genetic variants which kind of did the opposite to what we might expect. They were associated, versions of genes were associated with high body fat percentage, but kind of the opposite metabolic profile, so more good cholesterol, lower triglycerides, and more of these molecules here, which are traditional markers of body fat. And what this colorful slide shows is that in more detail, and they found 38 genes where a version of the gene had a traditional risk profile, where the red here is higher levels of body fat, higher levels of BMI, but blue is lower levels. See lower levels of HDL cholesterol, which is the good cholesterol, higher levels of triglycerides, the fat, etc. But you can see these 36 genetic variants here did very much the opposite to what you'd expect, in that the versions of the genes that go with higher body fat percentage and higher BMI, you actually have more good cholesterol and less fat in the blood amongst those other things. They have this opposite pattern of metabolic effects with the two types of genes. And our working hypothesis, our idea and aim was to really investigate whether people with the green type of genes, the favorable adiposity type of genes, had healthier looking fat cells. And we, again, from previous studies, suspected that these people would have healthier fat cells, and healthier fat cells tends to mean more but smaller fat cells. So you have more flexible or plastic, if you like, fat cells, whereas unhealthy fat cells tend to be fewer and larger. And you can think of this as, so you can see the diagram here, so you can think of this as unhealthy fat cells are being stretched. They're really having to store more fat than they want to, and so they end up being bigger, and that results in lots of other knock-on adverse effects. It means the surrounding tissue is more inflamed and doesn't function as well, and is generally not so good. So we hypothesized that we aimed to uncover whether people with different versions of the genes had healthier or unhealthier looking fat cells. And to do this there are actually two studies that did this. And the second one is in the newsletter, but the first one, which I'm going to talk about, is called FATDIVA, and we ended up recruiting 107 of you, 44 men and 63 women, and we decided to measure 36 of the favorable adiposity genetic variants, 36 versions of genes essentially that I mentioned in the previous slides. Basically give everyone a score between 0 and 72. And the score between 0 and 72 is basically adding up the number of versions of the favorable version of the gene. And because we can have two copies of any one gene, if you remember, one from mum, one from dad, if you have 36 versions of 36 genes you're looking at, you could have up to 72 favorable versions or 0 favorable versions. Most people are kind of in the middle, but there's variation. And what we did was, this is in many ways is a much simpler test. It was still involved your time and energy to come into the clinical research facility, and it involved a skilled nurse, Andy and Lindsay, Bea and others were able to do this. And so it was a skilled operation, but in many ways simpler than the glucose test I was talking about in the previous presentation. Because basically what we did was take a small pea-sized amount of adipose fat tissue from your abdomen, and we froze it, and then we, with the help of the histopathology department at the hospital, we made sections, sliced it very thinly, and made a microscope slide of that tissue, and we were able to look down the microscope and use some software to measure the size and number of the cells in any one particular view under the microscope, and that enabled us to measure the number, as I say, the number, area, and size of fat cells. And so again, I've split the results into two main sections: some general observations we found about the fat cells, and then secondly, how the fat cells related to our favorable and unfavorable genetic factors. And the first thing to say is that, again, very nicely a sort of positive control to our experiment is that we found some very meaningful correlations between your body dimensions, if you like, and the actual cells. So what this graph shows is the correlations between 1 and 0, so where 1 is a perfect correlation between the amount of, in this case, body fat percentage and the number of cells. You can see that there was about half the effect, basically. So your size of your fat cells was strongly correlated to the amount of to the percentage of your body fat to about half. BMI slightly less. Your non-fat mass, so this is your muscle and bone, etc., was not correlated at all, which was nice. And then the skin folds we took from behind your shoulder and around the back of your hips was also correlated with the number of with the size of your fat cells. Basically, the more fat however we measured it, the more cells you had. The larger the cells you had, which fitted with what we had expected. And then similar but slightly less strong correlations with the number of fat cells. So this is the critical bit where we equated the genetics with your size of your fat cells and the number of your fat cells. And just to walk you through this, in green at the top, we have the favorable adiposity genetics. And so the more cells you had, the more favorable adiposity genetics you had, the higher you were on that score of 0 to 72. That relates to what's happening to the size of your fat cells. And what you have here, sorry, I haven't got it annotated properly, but there's 3 bars, and the first bar is for everybody. The second bar here is for the men only, and the third bar is for the women only. And what this shows is that, and then these black bars here show the variation in the data, basically. And if that variation crosses this dotted line, that means there's no statistically meaningful difference. And so although you can see, particularly in the women, there's a trend towards the genetics, favorable adiposity genetics, resulting in smaller fat cells. It doesn't reach a meaningful level of statistical confidence. We also, I meant to say, we also did do this for the 38 genes which were associated with more traditional effects on metabolic factors. So the genetics that made you fatter also went with high risk of diabetes. And you can see things are, again, not statistically meaningful, but trending in the opposite direction. So especially in the women, that they tended to have larger fat cells than, although not really any effect in men. And everybody is kind of in between a bit. It got a little bit more interesting with the number of fat cells per slide, per cross-sectional unit, and that you can see this starts to become statistically more interesting in that especially the women, the more favorable adiposity genes they had, the more fat cells they had, and the more unfavorable adiposity genes or versions of genes they had, the fewer the fat cells they had. And so if you just put those 2 slides together, you can see, I think it looks quite reasonably convincing that, especially in women, that there are different patterns of fat cells based on your genetic factors, especially with the favorable adiposity genetic factors, resulting in basically more but smaller fat cells, and the unfavorable genetics resulting in fewer but larger fat cells. So I realize we are getting short of time. This is a common problem with scientists talking for too long. So that was, I think that's our most interesting result, especially in the women. Our next steps really are to try and follow this up in more detail, with more detailed adiposity measures and measures of fat cells, and the most importantly, to thank everybody who is in the Exeter 10,000 or yourselves for contributing your time and your insulin and your fat tissue to these studies. And another thank you especially to Michelle and Andy and Luke. There's behind these studies, there's a big team of people, both on the clinical side and in the scientific side, particularly Susie and Hanya, who've done much of the work. A real multidisciplinary collaboration between the scientists who are experts in the genetics and the clinical research facility who are expert in the tests and are experts in recruiting people. And it's something that Exeter does particularly well. There are, I think our Exeter 10,000 is one of the largest studies in the country that's able to do this sort of thing. So I will stop there. Thank you for your time, and very happy to take any questions. There's some nice complimentary comments coming through in the chat, but if you wish to raise any points, that is fine. We have a few minutes. Yes, I have many of you will have a small scar on your abdomen. You'll be pleased to know I also did this test. So Andy also stabbed me, and I have a nice little scar on my abdomen. My wife thinks I'm slightly mad. And also slightly mad for doing the test. Michelle said it'd be really great. I think she's going to send around an email. Obviously, online forums have their pros and cons. Often more people can join, but it's less interactive. In many ways, we will be happy to, I'm sure, at some point do a more in-person one if people desire. But any feedback about the format as well as the study would be gratefully received. Yeah, there'll be more information, particularly about the third study, which is closely related to the second study. Not exactly the same, but closely related. Is will be in the next newsletter, I believe. And if you read that, there's some of this should be familiar and help you. Are there any other housekeeping things? Michelle or Becky?
No, it's literally that we're going to be, Becky's going to email a link to a very short form to just get your feedback and any comments or suggestions you have. So thank you very much. If you can complete that, that would be great.
Okay. Thanks, Becky.
Thanks very much for everyone joining, and thanks very much to Tim for that great presentation.